Implantation is the embedding of a blastocyst into the endometrial stroma. It occurs after hatching of the blastocyst and during the implantation window. Hatching occurs when the blastocyst escapes and frees itself from the zona pellucida. Here you can see the blastocyst after escaping from the zona pellucida. It typically occurs about 1 to 3 days after the morula enters the uterine cavity. The implantation window is the time when the endometrium is maximally receptive. It typically occurs between day 16 and 22 of a 28-day menstrual cycle. It is 5 to 10 days after LH surge, and occurs between day 16 and 19 of a stimulated cycles by exogenous gonadotropin. Here you can see on this diagram the implantation window between day 16 and 22. The process of implantation can be divided into 3 stages: apposition, adhesion, and invasion. Starting with apposition, the blastocyst comes into contact with the endometrial epithelium. The blastocyst orients itself so the inner cell mass will point towards the endometrium. Pinopods and the glycocalyx play important roles during apposition. Pinopods are smooth microprotrusions from the apical uterine epithelial surface. They are present for a brief period of 2 to 3 days, occurring between day 5 and day 7 post-ovulation. They help in fluid absorption from the uterine cavity, attracting the blastocyst to the site of pinopods. This is an electron microscopic picture of pinopods, and when further magnified, it clarifies the shape of pinopods. Another important factor is glycocalyx. The uterine epithelium is coated with a network of glycoproteins called glycocalyx. It acts as a physical barrier to cell-to-cell interaction. The uterus is not receptive at this stage because of the thick mucin coat and the long microvilli. However, during the receptive stage, the microvilli shorten and the mucin 1 expression decrease in the immediate vicinity of the blastocyst. This image illustrates how mucin 1 acts as a barrier between blastocyst and the epithelial cell, and its removal allows interaction between the blastocyst and the receptors on the epithelial cells. Adhesion is the next step, when the blastocysts attach to the endometrial epithelium, and they are united together by the action of adhesion molecules. Key adhesion molecules include integrins, selectin, and troponin. The next step is invasion. After proper apposition and adhesion, the trophoectoderm start differentiation, forming cytotrophoplasts. These cytotrophoplasts are considered a trophoplast stem cells because they can further differentiate into either syncytial trophoplast or extravillous trophoplast. Cytotrophoplast will proliferate. The newly formed cytotrophoplast will initiate a cell-to-cell fusion, generating a cell with more than one nucleus. This multi-nucleated layer is called a primitive syncytial trophoplast. Syncytial trophoplast is able to penetrate the uterine epithelium and stroma. This allows the embedding of the embryo into the uterine stroma. During invasion, the primitive syncytial trophoplast comes into close contact with small capillaries and large uterine glands, and this provides nutrition for the newly developing embryo. Now the embryo becomes embedded inside the stroma. surrounded by syncytial trophoblast. Vacuolar spaces begins to appear within the syncytial trophoblast. These vacuoles will later coalesce together to form lacunae, which eventually will form the intervillous space. Cytotrophoblastic cells will proliferate inside the syncytial trophoblast, forming large finger-like projections. These are called villus cytotrophoblast, forming the primary villi. Subsequently, a layer of mesoderm will grow inside the primary villi to form them into secondary villi. At the tip of the villi, cytotrophoblasts will continue proliferation, forming columns of cytotrophoblast. Here is a secondary villus formed of outer layer of cytotrophoblast and a core of mesoderm. At the tip of the villus, cytotrophoblast will continue to proliferate, forming more layers of cytotrophoblasts, not invaded by mesoderm. These are called columns of cytotrophoblast, and they form the anchoring villi. These are the columns of cytotrophoblast. Then the cytotrophoblast will extend laterally to form what is known as cytotrophoblastic shell. This cytotrophoblastic shell represents the maternal-fetal interface, and it anchors the conceptus to the maternal endometrium. Initially, villi form around the conceptus in all directions, but eventually they regress in all areas except the area related to the decidua bizalis. I mean, these villi will regress at the end, and the only villi related to the decidua bizalis will remain and grow to form the placenta. At the tip of the anchoring villi, there is no covering syncytial trophoblast. A group of trophoblasts will leave the trophoblast cell columns and migrate into the endometrium and the upper part of the myometrium. These cells are called extravillus trophoblast. When extravillus trophoblast leaves the trophoblast cell column, they switch into an invasive phenotype. Extravillus trophoblasts are further subdivided into endovascular and interstitial trophoblast. Endovascular trophoblast invade and transform the spiral arteries, while interstitial trophoblast invade the decidua and surround the spiral arteries. This eventually will lead to remodeling of the spiral arteries. It converted from a narrow lumen muscular spiral artery into a dilated, low resistance uteroplacental vessel. And about 1 month after conception, maternal blood will enter the intervillous space. The interstitial trophoblast invades the decidua and eventually penetrates the myometrium to form a placental bed giant cells. They also surround the spiral arteries. On the other side, the endovascular trophoblast first they enter the spiral artery lumen and initially form a cellular plug. Then they destroy the vascular endothelium and modify the vascular medium. And finally, a fibrinoid material will replace the smooth muscle and the connective tissue of the vessel media. The spiral arteries later will regenerate endothelium. Invading into vascular trophoblast can extend several centimeters along the vessel lumen, and they must migrate against arterial flow. However, invasion by trophoblast involve only the decidual spiral arteries and not decidual veins. This spiral artery remodeling occurs in 2 waves. The first wave occurs before 12 weeks, post-fertilization, and the spiral arteries are invaded and modified up to the level between the decidua and the myometrium. The second wave occurs between 12 and 16 weeks, and it involves some invasion into the intramyometrial segment of the spiral arteries. And finally, there are several mechanisms involved in controlling invasion. One factor is the formation of a Nitabuch layer. It is a fibrinoid layer formed at the maternal-fetal interface. It prevents excessive penetration of the trophoblast into the uterine wall. It also creates a physical separation between the maternal and the fetal blood circulation, preventing immunological rejection or infection. Another factor that can limit invasion is the secretion of various interleukins, growth factors, and other factors by decidual cells. These factors can be classified as either supporting invasion or inhibiting invasion. Thank you.